You begin by feeling the delicate threads of life, spiraling and coiling like a gentle river of information within every cell. It is a quiet symphony, each nucleotide a note in a melody composed long before you were born. You sense the rhythm of adenine reaching for thymine, cytosine pairing with guanine, forming the soft, precise patterns that have guided life across countless generations. This is a world both intimate and immense, tucked inside you yet vast in its echoes of ancestry. As you drift closer, your awareness expands into the microscopic architecture of the nucleus. Chromosomes rest in their organized clusters, like ancient scrolls, each wrapped in a protective embrace of proteins, keeping secrets yet to be fully discovered. Your mind moves along these delicate coils, noticing how they bend, twist, and fold. A choreography both orderly and organic. It is here, in the silent dance of DNA, that life's instructions are whispered, not shouted. You trace a single strand, imagining it as a winding river of light and shadow. The sugar-phosphate backbone hums softly under your fingertips, a structure holding together countless sequences, like bridges spanning endless streams of possibility. Along its path, the bases rise and fall, forming the letters of a language older than any spoken word. You feel the elegance of this code, its resilience, its flexibility, capable of both preserving history and guiding transformation. Your attention drifts to the replication process, a gentle echo of continuity. Imagine the strands slowly unwinding, unzipping in delicate symmetry. Each base finds its partner, perfectly matched, as if nature itself whispers the answers before you even ask the questions. The polymerase enzymes move with patient diligence, weaving new strands with quiet fidelity. It is not hurried, yet there is certainty, a timeless rhythm ensuring that every new cell carries the essence of the one before. You notice mutations as soft as a sigh, tiny changes that ripple through the sequence. Some drift unnoticed, others shape new possibilities. They are the gentle brushstrokes of evolution, subtle hints that life is not static but always unfolding. You let your mind float alongside these variations, sensing the delicate balance between stability and change, the quiet creativity that emerges from imperfection. In this meditative journey, you consider how DNA is more than code. It is a library of stories, a repository of experiences, written in molecules that bend and fold like ribbons in a gentle breeze. Each gene is a stanza, each regulatory sequence a pause, shaping the melody in ways both visible and hidden. You feel a profound intimacy with the strands, a connection that bridges time, linking you to ancestors and descendants alike, and to every organism whose life depends on this same molecular script. You notice the molecular machines at work, helicases unwinding strands, ligases sealing gaps, topoisomerases smoothing the twists. Each movement is precise, fluid, almost musical in its coordination. The cell is a concert hall, every molecule playing its part with care and attention. You sense the quiet intelligence embedded in these interactions, not conscious, but profoundly ordered, a testament to the subtle artistry of life. Your thoughts float to epigenetics, the gentle touch that does not rewrite letters but adjusts the volume of the story. Methyl groups, histone modifications, and chemical marks act like soft brushstrokes on parchment, highlighting certain passages while leaving others muted. Life, you realize, is not only written in the sequence of bases but in the way the script is read, interpreted, and experienced. You feel the adaptability, the quiet dialogue between environment and code, as though the cell itself listens, senses, and responds. You wander into the world of non-coding DNA, once thought of as silent or meaningless. Here are the whispers, the hidden conversations of the genome. Long RNAs fold into intricate shapes, guiding molecular processes, shaping the timing of gene expression, forming networks of subtle influence. These regions are quiet yet essential, a reminder that life's complexity often resides in the gentle, almost invisible interactions, as much in absence as in presence. In the mitochondria, you sense another strand of DNA, smaller, circular, carrying a story of energy and endurance. It hums with the rhythm of metabolism, the quiet pulse of cellular respiration. Here, life's power flows from molecule to molecule, fueling the processes that allow the genome in the nucleus to rest, replicate, and whisper. You feel the duality of control and cooperation, two strands of life speaking in harmony, balancing continuity and vitality. You drift deeper into reflection, thinking of DNA as time's vessel. Each cell is a microcosm, each nucleus a library, each strand a record of what has been and a hint of what might be. There is a sense of patience, a slowness that soothes. Life does not rush, it evolves, it unfolds, it listens. You feel your own body in this rhythm, sensing the quiet pulse that matches the molecular symphony, your breath in sync with the gentle spirals of coiling strands. You notice the universality of this language. From bacteria to humans, the same bases form the same pairs, the same structures give rise to life in forms unimaginably diverse. You feel connected to every living thing, a thread extending outward through time and space, woven together by the soft, persistent whisper of DNA. Each organism carries this melody differently, yet all are variations on a theme, unique interpretations of the same molecular song. As your mind follows these delicate threads, you sense the hidden elegance of DNA repair. Enzymes slide along the strands, correcting errors, smoothing irregularities, ensuring that life's story continues without disruption. There is no haste, no judgment, only gentle correction, the quiet attention of molecular caretakers preserving the integrity of life. You feel gratitude for these invisible processes, the silent guardians of continuity. You imagine the entire genome as a landscape, valleys and peaks, forests of genes, rivers of regulation, all interacting in a delicate balance. In this landscape, nothing is wasted. Every part has a role, every interaction a purpose. You wander gently through this terrain, letting your mind rest in the harmony of natural order, feeling the soft rhythm that underpins existence. Your awareness begins to expand outward, leaving the nucleus but carrying its essence with you. You feel the thread of life extending through your body, through generations, through the invisible network of ancestry. It is a calm, enduring presence, a whispering guide, a soft reminder of the continuity that connects you to all living things. And as you linger in this quiet reflection, you sense that the story is only beginning. The genome has many tales to tell, and you are about to explore the next, where these threads weave into the larger fabric of life itself, revealing how the whispers of DNA orchestrate the symphony of existence. You step closer into the heart of the garden, where light dances through leaves of DNA, folding and twisting in patterns that feel almost musical. Here, the air carries a gentle hum, a vibration of life that is quiet but insistent. You notice a new presence, subtle yet unmistakable, moving along the winding strands like a careful gardener tending an endless maze. This is Crispr, though it does not announce itself with fanfare. Instead, it moves with patience, guided by something deeper than instinct, a sense of purpose embedded in code. At first, you cannot quite see it. It is smaller than your mind can easily imagine, smaller than any creature you have ever observed. It exists in the spaces between molecules, a delicate hand reaching for threads of genetic material. The garden itself seems to lean toward it, allowing it access to certain leaves, certain vines, while leaving others untouched. You watch with quiet awe as the tiny molecular scissors glide along a DNA strand, pausing here and there, cutting with precision that feels almost sacred. Crispr's movements are slow, deliberate. You feel a sense of intimacy, as though the gardener is whispering to the garden, asking each plant if it is ready for transformation. There is no violence here, no abrupt force, only gentle guidance, subtle rearrangement, and a rhythm that you begin to sense in your own heartbeat. You realize that each cut, each edit is not merely a correction. It is a conversation between what exists and what could exist. You remember learning that Crispr was first discovered in bacteria, simple creatures with no consciousness, yet somehow carrying the seeds of this incredible tool. In their world, Crispr is a memory, a record of past viral invaders, a way to defend against threats with elegant efficiency. And now, in your hands, through your imagination and through careful human study, it has become a key to understanding life on a broader scale. You feel the quiet thrill of recognition, a mechanism so tiny, yet so profound, capable of shaping the very code of living things. The scissors hover above a strand, guided by a tiny RNA molecule. You think of this RNA as a gentle pointer, a note left on the blueprint, saying, "This is where attention is needed." It drifts softly, finding its match, allowing Crispr to engage only where it is needed. You feel a wave of clarity as you realize that there is intention even at this microscopic level, a principle of organization and purpose running through the chaos of the cellular world. You begin to imagine what it would feel like to be inside the cell itself, moving alongside these tiny instruments. The DNA strands are not cold or rigid. They pulse with energy, curling and stretching in a dance that is both ancient and ever new. You see sections that fold back on themselves like petals, sequences that loop in on their own histories. And through it all, Crispr moves with gentle authority, trimming a leaf here, nudging a vine there, guiding the garden toward a subtle, deliberate harmony. There is a rhythm to this process, a pulse that is almost musical. You notice that after each precise cut, the DNA tends to shift slightly, settling into a new position as if relieved to be guided. The garden itself breathes more easily, releasing tension that you didn't realize had existed. You feel your own body mirror this rhythm, your breath slowing, your mind softening. Curiosity melts into wonder as you follow each movement, each small transformation, with quiet devotion. You recall how humans have learned to harness this tool, to carry the patience of bacteria into our own understanding. Crispr has become more than a molecular gardener. It is a mirror of our own curiosity and care. It reminds you that the smallest actions, guided by understanding and attentiveness, can ripple outward, reshaping entire systems without chaos or haste. You are humbled by the precision and by the quiet persistence of life itself. Sometimes Crispr pauses, hovering over a section it has already visited. You imagine it listening, as if consulting the garden itself, asking, "Is this enough? Is this correct?" You realize that even in a mechanism so precise, there is room for reflection, for gentle assessment. The notion surprises you. Science, often thought of as rigid and exact, here feels soft, meditative, alive. The garden, the molecular tools, and you exist together in this shared space of attentive transformation. You notice that each edit carries consequences far beyond its immediate vicinity. One small cut may influence the growth of a nearby strand, the behavior of a neighboring cell, the rhythm of an entire pathway. You feel the responsibility of awareness pressing gently against your mind. Life is intricate, delicate, and yet remarkably resilient. Crispr moves with respect for this resilience, making changes that are measured, thoughtful, and elegant. As you watch, you start to see the beauty of mistakes and corrections, of trial and error encoded in the strands themselves. Even errors are part of the process, teaching the gardener where adjustments are needed, where patterns require a softer hand. You feel a profound kinship with this microscopic patience, this attentive editing, and the realization settles into you. Transformation does not require force. It requires care, observation, and gentle guidance. In this quiet garden, time stretches differently. Minutes become expansive, filled with the deliberate unfolding of tiny interventions. You imagine watching Crispr in a slow motion that reveals hidden choreography, each movement a gesture of balance, each pause a reflection on harmony. You feel gratitude for the discovery of such a tool, for the way it allows insight into the most intimate processes of life without shattering their mystery. And yet, as you linger, you sense the limits of what you see. Crispr works invisibly, inside countless cells across infinite possibilities. Its presence is real but subtle, tangible in effect but elusive to the eye. You feel a soft wonder at the paradox. A force so small, so patient, capable of shaping the blueprint of life itself, yet so quiet that it could almost be mistaken for stillness. You lean closer, imagining your fingers brushing against a DNA helix, and feel an unexpected warmth, a connection to the living world that is both scientific and poetic. Every edit, every precise cut, is a reminder of the intimacy of discovery, of the gentle art of understanding life from within. You realize that Crispr is not just a tool. It is a teacher, showing the patience required to observe, to intervene carefully, to nurture the unfolding story of life. As the sun seems to lower itself over this garden, you notice a faint glow along the DNA strands, illuminated by the quiet diligence of the tiny gardener. You feel a wave of calm wash through your mind. Curiosity has softened into reflection, into awe, into a kind of gentle companionship with life itself. And in this soft quiet, you sense that the garden has many more secrets, waiting for a patient touch, waiting for attention and care, waiting for the next story to unfold in the DNA skies above. A thought rises softly in your mind. If a tiny pair of molecular scissors can guide and reshape an entire garden, what wonders might patience, care, and curiosity reveal in the rest of the living world? You drift closer, and suddenly, the world around you shrinks. The air seems lighter, the space more intimate, and all at once, you are no longer in your familiar room, but in a vast, invisible landscape teeming with life. Bacteria move with a quiet purpose, their motions gentle yet deliberate. They inhabit droplets of water, clinging to surfaces, thriving in places too small to see, too subtle to hear. And in their movements you sense lessons written not in words but in patterns, in choices, in the pulse of survival itself. You watch as a single bacterium divides, splitting perfectly into two, a mirror of life giving birth to life. The process is graceful, almost ceremonial, and you realize that every division carries a story of persistence. Each cell is a tiny vessel of memory and experience, holding within it the strategies of generations past. Evolution whispers here, softly, teaching patience, ingenuity, and resilience. Nearby, you notice clusters of bacteria forming thin layers, glistening like dew upon leaves. These biofilms are more than just protection. They are communities built on cooperation. Cells share nutrients, exchange chemical messages, and sometimes even surrender themselves to serve the whole. You feel the gentle hum of this collective intelligence, the soft rhythm of collaboration. It reminds you that even the smallest beings find ways to support one another, creating resilience through unity. A subtle vibration passes through the environment, a signal carried in molecules drifting like whispers on the wind. This is quorum sensing, a chemical conversation that coordinates the behavior of thousands of individual cells. You lean closer and for a moment you can almost understand. There is a pattern to their dialogue. Warnings, celebrations, and strategic decisions unfolding invisibly, shaping the survival of the community. You sense how communication is essential, how even unseen words can alter the course of life. Some bacteria you observe cloak themselves in layers of protective armor. Capsules, spores, and hardened walls form a quiet fortress, guarding against threats you cannot see. It is an ancient wisdom, perfected over eons. Survival often depends not on brute strength but on preparation, foresight, and subtle ingenuity. You notice how these defenses are tailored to their environment, elegant solutions honed by trial and time. Every barrier, every shield is a lesson in balance, a reminder that protection can be as much about restraint as about action. In the corners of this microcosm, extremophiles thrive, unbothered by heat, cold, acid, or scarcity. They flourish where life seems impossible, rewriting the rules of survival. Watching them, you feel a quiet awe, the realization that life is adaptable in ways you might never imagine. There is courage in persistence, and ingenuity in resilience. Even in the harshest conditions, the smallest forms of life find a path forward, carving meaning from adversity. Occasionally, you see bacteria exchanging fragments of DNA. This horizontal gene transfer is a subtle form of teaching, a passing of lessons from one generation to another, or even across species. It is a delicate exchange, almost intimate, carrying adaptations and strategies that could change the fate of entire populations. You sense that learning is everywhere in this micro-world, written not in textbooks but in living code, silently guiding the next wave of life. As you observe, patterns emerge. Cycles of growth and rest, cooperation and competition, attack and defense. Life here is not chaotic. It is a careful, measured dance. Each decision, each adaptation carries weight. Every tiny action resonates through the invisible ecosystem, echoing in ways that are both profound and gentle. You begin to understand that survival is not simply about avoiding death, but about responding, adjusting, and coexisting. You notice that these microbial strategies reflect lessons that humans can appreciate: resilience, adaptation, communication, and collaboration. Each tiny organism offers insight into problem-solving, into thriving despite limitations, into the quiet ingenuity of life. You feel a soft connection between your own experiences and the microscopic world, as though the rhythm of evolution beneath your thoughts mirrors the challenges and adaptations in your own life. The bacterial universe hums with gentle wisdom. Even in its invisibility, it speaks clearly about persistence, ingenuity, and the quiet power of small actions. You linger a moment longer, letting the stories of these tiny beings wash over you, and you realize that the world is richer, more connected, and more alive than it seems. Their lessons are subtle, but they are there, waiting to be noticed, waiting to inspire. And as you prepare to drift from this microscopic world, a single thought lingers: that every adaptation, every choice, is a quiet testament to life's persistence, and that there are still countless secrets hidden in the invisible currents of survival, waiting to meet your curiosity. You approach the strands of DNA with reverence, each molecule a quiet corridor of instruction, a scroll of life written in a 4-letter alphabet. The letters, adenine, thymine, cytosine, and guanine, line up with precision, forming words and sentences that shape the forms and functions of all living things. Today, your focus narrows to the spaces between these letters, the tiny moments where change can ripple outward and alter the course of life. You sense the intimacy of this process, the weight of responsibility that comes with touching the very fabric of existence. Crispr appears before you, not as a cold tool, but as a delicate brush in the sunlight. You watch as it glides along the helix, searching for a precise sequence, a string of letters that may need a gentle nudge. The molecule is guided by RNA, a mirror that knows exactly where to look, as if remembering the contours of the DNA like a friend knows the rhythm of your heartbeat. There is patience in this pursuit, a quiet focus that reminds you how even the smallest adjustment carries meaning beyond imagination. You feel the paradox of scale. On one hand, the edits are microscopic, mere molecules shifting in careful choreography. On the other hand, the consequences are immense. A single change can influence growth, resilience, and adaptation. You sense the responsibility that accompanies such power. Each base you touch is a choice, a meditation on possibility, a conversation with the future. You imagine scientists, like quiet gardeners, pruning and shaping life with both caution and creativity, aware that their touch must be precise, intentional, and compassionate. The process unfolds slowly before your mind's eye. Cas9, the enzyme that acts as Crispr's hand, glides along the strand, scanning, pausing, poised with patience. You imagine it pausing at the correct site, aligning perfectly with the target sequence. Then, with a gentle incision, it opens the helix, like a flower revealing its hidden center. You feel the intimacy of this act, the precision required to make one small cut without disturbing the entire structure. It is a meditation in motion, a choreography of molecules performed without haste, with grace and care. You notice how the RNA guide serves as a quiet teacher, pointing the enzyme to exactly where change is needed. The system is both elegant and humble, a demonstration of nature's efficiency combined with human ingenuity. There is rhythm in the process, a wave-like movement that mirrors your own breath. You inhale the thought of potential, the possibility of correction, the restoration of function. You exhale the recognition of risk, of complexity, of the care required to respect the integrity of life while making adjustments. Your awareness drifts to the philosophical weight of editing life. Each edit carries echoes into the future. A correction in a single cell may expand to influence generations, each ripple touching countless lives in subtle or profound ways. You feel the interconnectedness of all existence, the fragile balance that life maintains, and the profound responsibility that comes with participating in this ongoing story. You are a witness to creation and modification, an observer of the quiet interplay between power and humility. The letters shift before your mind's eye. Adenine pairs with thymine, cytosine with guanine, and yet in the spaces where Crispr intervenes, new sequences emerge. You imagine these changes as gentle brushstrokes on a canvas of living potential. Some edits restore harmony where there was error, others introduce subtle variation that may ripple into unforeseen creativity. There is beauty in the uncertainty, in the dance between control and discovery, in the balance between intention and the natural unfolding of life. You sense the care required to measure each action. The genome is not merely a list of instructions. It is a library of possibilities, a map of history, a record of evolution, and a blueprint for the future. You feel the enormity of the responsibility, the gentle humility required to touch even a single letter. Each choice is a meditation, each pause a moment of reflection. You notice how the smallest edits can echo in ways you cannot predict, like a single stone dropped into a vast, calm lake, rippling outward in concentric circles of potential. As Crispr moves along the strand, you sense an almost musical rhythm. There is anticipation, alignment, incision, repair, a quiet symphony of molecular motion. You imagine DNA repair enzymes flowing in like gentle currents, knitting the strand back together, ensuring continuity and balance. You feel a connection to this natural choreography, sensing how life maintains equilibrium even when touched by innovation. The process is at once intimate and expansive, a reflection of the universe's subtle balance. You reflect on the philosophical implications of editing life. There is potential for healing, for correcting errors that cause suffering, for understanding and shaping adaptation. Yet there is also the need for restraint, for respect, for wisdom in knowing when not to intervene. You feel the delicate tension between power and humility, the meditation of responsibility, the quiet awe at the elegance of a system that allows both precision and creativity. Each thought slows your mind, guiding it toward reflection, toward appreciation for the profound interplay of control and surrender. You observe the changes ripple through the strand like gentle waves. A single base is replaced, a small mutation corrected, a tiny adjustment made with care. Yet the impact is larger than it seems. Proteins fold differently, cells function more smoothly, possibilities emerge that were once hidden. You sense the beauty of this delicate architecture, how each part of life depends on countless invisible interactions. You feel gratitude for the precision and elegance of molecular machinery, for the intricate choreography that allows life to continue with grace. The process is both intimate and meditative. You are present in every incision, every pairing, every repair. You notice the quiet patience required to achieve balance. You sense the humility of the scientists, the wonder of the molecules, the endless creativity of evolution. Each edit becomes a meditation on possibility, a reminder that change is both a gift and a responsibility. You let yourself linger in this awareness, letting your mind breathe, letting your imagination embrace the gentle intricacies of molecular life. You feel the tension between intervention and observation, between curiosity and care. Crispr is a tool, a brush, a hand reaching gently into the past and future of life. You imagine it poised above a single base, contemplating its action, aware of the vast consequences that may follow. The moment is quiet, almost sacred, a meditation on power, responsibility, and the infinite potential contained within the tiniest unit of life. Finally, as the edits settle and the molecule folds back into its familiar form, you sense completion without finality. Life continues to hum, quietly and gently, carrying with it the echoes of your reflection. The process leaves you with a sense of wonder, a sense of responsibility, and a soft, lingering awareness that every choice, every edit, carries both potential and humility. You close your eyes and let your breath settle into the rhythm of your body. Imagine the quiet hum of life within you, cells working in harmony, each carrying a carefully written instruction, a guide to what you are and what you will become. These instructions are the genetic codes, long strings of letters, quietly folding and unfolding to shape the very essence of who you are. They are intricate, delicate, and in some ways, like a manuscript, capable of holding the beauty and the burden of mistakes. Sometimes, these instructions contain errors, little typos in a vast manual. Some are small, barely noticeable, barely disturbing the flow of life. Others are larger, more disruptive, creating discomfort, pain, or disease. But now, there exists a quiet hope, a gentle science that does not shout or demand, but nudges and corrects. You imagine Crispr, a word that sounds like the soft rustle of leaves, yet carries the precision of a surgeon's hand, moving delicately along these genetic threads. Crispr is a tool, a pair of molecular scissors guided by the wisdom of natural bacterial defense mechanisms. In your mind, you see it floating among your cells, seeking out sequences that need attention. The process is methodical, almost meditative. The scissors locate the precise letters that are miswritten, and a new sequence, accurate and harmonious, is offered in its place. It is as if a tiny librarian moves quietly through the library of your DNA, carefully replacing a worn page with one that restores clarity to the story. You feel a sense of relief just imagining it. The errors that once caused distress can now be softened, rewritten, or silenced. The therapy is not invasive in a way that shocks your system. It flows gently, working from the inside out, harmonizing rather than forcing. It reminds you of the way sunlight gradually melts frost, or how a soft tide reshapes the sand. You do not resist, and you do not hurry. Everything unfolds at a natural, deliberate pace. As you drift further into this imagery, you notice the quiet intelligence behind these interventions. Every cut and correction is guided not by randomness, but by understanding, by observation, by the careful design of those who have studied these sequences for years. They know that each gene carries not just function, but memory, history, potential. To change one is to alter a ripple through the body, a subtle adjustment in a complex system. And yet, the care is present, the precision unwavering, allowing life to continue its course with newfound support. You imagine the patients who might one day experience this. Children whose bodies carry burdens they did not choose, adults who have endured years of imbalance, all meeting a gentle form of science that does not overwhelm. The hope is tangible, like a soft warmth in your chest. With every imagined cell repair, you sense the possibility of release, relief from suffering, freedom from limitations, a quiet restoration of equilibrium. It is not magic. It is meticulous, thoughtful intervention, the result of observation, experimentation, and understanding. And yet, you also feel the humility of this process. Life is intricate, and no correction comes without contemplation. Scientists move slowly, respecting the delicate balance that has evolved over millennia. Each change is considered carefully, knowing that while relief can be profound, the body systems are intertwined, and unintended consequences must be watched for with patience and care. In this mindful approach, there is a lesson for you. That true healing, whether in science or within your own life, comes from gentle attention and thoughtful action, not force or rush. The potential of Crispr is also a reminder of connection. You realize that your genetic story is shared with countless others, that each correction has echoes in the tapestry of life. By mending a broken instruction in one body, you participate in a quiet chorus of restoration, a collective movement toward balance. You are part of a continuum, where human ingenuity meets the natural intelligence of cells, and where hope is not a fleeting wish but a careful, deliberate practice. In your mind's eye, you see the therapy moving through the bloodstream, a calm, deliberate tide carrying promise to distant cells. It slips into nuclei, delivers guidance, and steps back, allowing the cell's own machinery to complete the work. There is no violence, no sudden shock, only the subtle, patient reshaping of what was once imperfect. You feel a soft gratitude for this unfolding process, a recognition of the quiet diligence that makes life better without fanfare. You notice the metaphor of a garden unfolding. Some seeds are planted, some sprout with difficulty, and others thrive effortlessly. Where growth is hindered, gentle intervention can guide, prune, and nourish. Crispr acts like a careful gardener, not forcing blooms, but creating conditions in which life can flourish naturally. You breathe in the image, letting it settle into the back of your mind, and you notice how it mirrors your own life. That small, thoughtful corrections and attentions can create peace, growth, and harmony without strain. And so, you imagine the broader picture. Not only rare diseases but common ailments, subtle genetic predispositions, can be approached with this same gentle care. The potential feels vast, yet it is tempered by the respect for complexity. The mind quiets in recognition that knowledge and patience together can create something profoundly soothing. A world in which biology and understanding work together in rhythm, like a slow, deep breath. You feel the intimacy of this knowledge. Inside you, every cell is a universe, and inside each universe, tiny interventions can have resounding effects. You sense the quiet beauty of existence, of molecules aligning, instructions being corrected, life finding a balance that was previously hidden. You rest in this gentle possibility, imagining a body not at odds with itself, but in conversation with its own healing potential. As the imagery softens, you sense the transition waiting beyond this moment. Genes can be rewritten, cells can be nurtured, and equilibrium can be restored. But beyond individual repair lies the larger wonder of how all systems interact. The way one intervention ripples into the interconnected web of life, the brain, the heart, and the mind. You realize that just as Crispr moves carefully through DNA, so too does curiosity and understanding move through the universe of knowledge. And here, in this gentle reflection, you allow your awareness to drift toward the next layer of discovery, where healing is not only internal but also shared, where the quiet intelligence of life reaches beyond the microscopic into the expanses of thought and exploration, hinting at the next journey you will take in this meditative exploration. You wander into a garden unlike any other, a place where possibilities grow like delicate stems reaching toward the sun. Each plant is unique, patterned with the quiet logic of life, yet shaped by forces both natural and deliberate. In this garden, you begin to notice the subtle touches of human hands intertwined with the soil, not harsh or abrupt, but gentle, as if someone has whispered suggestions to the seeds before they germinated. You pause at a cluster of small blossoms, their colors shifting with the light, each hue a reflection of possibility. Here, life is malleable in ways that were once unimaginable. Science has given humanity a toolkit, a palette, to shape the earliest whispers of being. And yet, in the soft breeze, you sense the weight of choices, like shadows stretching quietly across the garden path. You think of genes as threads in a tapestry, each one carrying a story from generations past. Some threads are strong, others fragile. Some bring resilience, others vulnerability. In the hands of a careful gardener, these threads can be woven into new patterns, strengthening the tapestry or introducing new designs. You wonder what it means to weave deliberately. When is it an act of care, and when does it tip toward a subtle form of control? A small pond reflects the sky above, rippling with every gentle movement. You see your own face mirrored there, but it is accompanied by countless reflections of potential selves, possibilities that exist only in the quiet promise of the water. The pond reminds you of the unknowns in this ethical garden, the outcomes that cannot yet be predicted, the long-term effects that unfold slowly, like water eroding stone over centuries. In the stillness, you notice that even small changes can resonate far beyond their point of origin. You walk along a winding path lined with trees, their branches bending lightly under the weight of unseen fruit. Some fruits are familiar, like apples or pears, representing the known benefits of scientific intervention. Cures for genetic diseases, the relief of suffering, the gift of time and health. Others are strange, shimmering with hints of colors that do not yet have names, mysterious hybrids that spark both wonder and hesitation. They symbolize the choices that carry more uncertainty, the subtle redesigns of life that could ripple outward in ways no one fully understands. You pause under one particularly luminous tree, its leaves catching the sunlight in a way that makes each one glow as though it contains its own heartbeat. Here, you consider the principle of intention. The gardener who shapes these lives must act with mindfulness, not haste, with curiosity, not arrogance. You feel the gravity of this quiet responsibility. To intervene in the code of life is to step into a conversation that stretches across time, across generations, across the invisible web that connects all living things. A soft wind lifts petals into the air, and you watch them drift lazily, sometimes landing gently, sometimes carried away. You notice how small choices, almost imperceptible at first, can influence the path of growth. A single decision to alter a gene, to prevent a disease, to enhance a trait, is like releasing a petal into a stream. Its direction cannot be fully controlled, yet it carries both intention and consequence. You feel the delicate tension between possibility and humility, between curiosity and reverence. You wander further, passing a section of the garden where wildflowers grow unchecked, vibrant and unplanned. They remind you that not all beauty is designed, that nature has its own rhythms and wisdom. Here, intervention must be measured, a gentle conversation rather than a command. You notice how even the most well-intentioned gardener must pause, must observe, must listen. The garden teaches patience, the kind that allows space for life to reveal its complexity without rushing to judgment. A narrow path leads you to a small bridge over a quiet stream. Leaning on the railing, you watch water ripple over smooth stones, each curve a reminder that life unfolds in layers and flows. You think of science as a current. It moves, shaping the banks, sometimes gently, sometimes more forcefully, but always in motion. The question arises, how do we navigate this flow responsibly? How do we participate without forcing direction? You sense that the ethics of the garden are not rules to follow, but questions to carry with care, like stones in your hand, feeling their weight and texture. In a shaded corner, you find a bench beside a grove of young saplings. Their shoots stretch upward, tentative yet full of promise. You sit and reflect on the principle of consent in this quiet space. Life does not ask for permission in the way humans do, yet the choice to alter it carries moral weight. Each sapling is a life in potential, a story that has not yet begun. How do you honor its unfolding, while gently guiding it toward health, resilience, and possibility? You notice your breath slowing, your awareness deepening. Questions and wonder coexist, neither demanding immediate answers, simply inviting presence. Above, the branches form a canopy, sunlight filtering through in dappled patterns, reminding you that knowledge itself is light, illuminating some paths while leaving others in soft shadow. You notice the interplay of clarity and uncertainty, how one cannot exist without the other. Science provides insight, yes, but it is paired with the humility to recognize what is unknown. In this garden, ethics are woven into every choice, every careful touch, every observation. In the same way that sunlight and water nourish all growth. You imagine walking among these living threads, guided by curiosity and respect, noting the subtle balance between intervention and letting be. Each plant, each blossom is a story. Some are written in resilience, others in fragility. Some bear the marks of thoughtful design, others the grace of unplanned wonder. In the quiet, you feel your own reflection mirrored in these choices, and a soft awareness settles. That participation in life's unfolding is a delicate art, a meditation of responsibility and care. Slowly, you rise from the bench, stepping back onto the winding path. You notice the way the garden seems to exhale with you, leaves trembling lightly, petals turning in acknowledgement. Here, there is no urgency, no need for immediate resolution. Only the gentle invitation to observe, to ponder, to hold questions with calm curiosity. And as you walk forward, you carry the soft, unspoken understanding that the ethical garden is not a destination, but a living, breathing conversation, one that stretches beyond your sight, beyond your years, and into the quiet possibilities of what life might become. You step back, letting the human horizon soften, and breathe in the world that exists beyond your immediate knowing. You feel the warmth of sunlight on leaves, a touch so gentle it seems to pulse in rhythm with your own heartbeat. There is a quiet complexity in the dance of roots beneath the soil, weaving networks that connect trees, shrubs, and grasses into conversations you cannot hear but can sense. Tiny insects hum along the threads, carriers of messages too subtle for your eyes, yet essential to the fabric of life. You notice the way the wind moves through the canopy, shaping leaves and bending branches in a slow, deliberate choreography, each motion part of an intricate, ongoing symphony. Your attention drifts toward the streams that thread through the landscape, ribbons of life reflecting sky and earth. Fish trace the currents, glinting silver in soft arcs. Each movement nudges the water slightly, sending ripples outward, reaching stones and reeds in patterns as old as time. You realize the world is alive not only in the organisms you see but in the connections they form. Invisible, persistent, and infinitely patient. Even the moss clinging to shaded rocks participates in this web, quietly filtering water, exchanging nutrients, and shaping the microhabitat around it. Everything pulses with purpose, yet it is subtle, almost imperceptible unless you slow down enough to perceive the delicate balance. In this expanded horizon, humans are not the center but participants, curious, careful, and responsible. You feel the weight and the wonder of knowing that your interventions, no matter how well-intentioned, ripple outward. Science becomes a lens to see these threads, a tool for understanding and stewardship rather than dominion. Crispr, with its precise edits, enters this consciousness not as a weapon, but as a paintbrush, gentle enough to correct yet mindful enough to preserve. Each nucleotide you imagine altering sends waves through a network of unseen consequences. Like touching the water with a single finger, you watch the concentric rings extend beyond what you could immediately foresee. The elegance of this biological network feels poetic. You consider the pollinators, bees, butterflies, birds, even the hum of tiny nocturnal moths. Their journeys are mapped across petals, leaves, and fruits, touching blossoms in a choreography older than memory. Each visit carries pollen. Each departure promises life to the next bloom. You realize these small actions sustain not only plants but entire ecosystems, allowing herbivores to thrive, predators to flourish, and the soil itself to remain fertile. You see how CRISPR might assist in this dance, perhaps rescuing a fragile species from disease, or restoring genetic diversity where it has been lost, but only if each action is measured, intentional, and respectful of the web it inhabits. Your mind wanders to the forests, where sunlight filters through branches in slow, liquid streams. Leaves shimmer as they shift, a dialogue of green and gold. Roots beneath intertwine like the lines of an ancient manuscript, exchanging sugars, signaling threats, and supporting one another in unseen ways. You understand that every action humans take, even in distant labs, echoes here. The ripples from a CRISPR edit in one gene might affect microbial communities in the soil, the insects that feed on those microbes, and the birds that eat those insects. You imagine following each concentric circle outward, noticing its subtle touch on life that does not belong to you, yet belongs to the network you now inhabit in thought and attention. Stepping closer to the animal realm, you notice how behaviors emerge in patterns, a subtle choreography that sustains the whole. Ants build their colonies with a silent understanding of structure, resource, and need. Wolves hunt with precision, not cruelty, following instincts honed over generations. You see the connections. Predators regulate populations, prey ensure soil enrichment, scavengers recycle energy, and decomposers dissolve death into the materials of new growth. Each action, small or monumental, reverberates through the ecosystem like chords in a vast symphony. The hum of life continues even when humans are unaware, and your awareness of these ripples becomes a meditation itself, a soft acknowledgement of humility and awe. Now, consider the implications of applying human knowledge here. Genetic tools like CRISPR are capable of creating change that is elegant, targeted, yet profound. You imagine restoring a coral reef, strengthening its resilience against warming seas by gently nudging the genetics of its polyps. You picture editing a crop to resist a disease without removing the microbial partners that nourish its roots. Each hypothetical intervention carries a lesson. Wisdom must accompany ability, observation must guide action, and patience must temper desire. You feel the weight of responsibility, yet also the grace in knowing that careful, mindful steps can resonate through generations of life. You take a moment to focus on the silent, subtle creatures often overlooked, the soil microbes, fungi, and bacteria. They are the unseen architects, decomposing, cycling nutrients, shaping water flow, and forming symbiotic alliances with plants. The networks they create stretch across landscapes, connecting trees in ways we are only beginning to understand. Your imagination traces the pathways of molecules and electrons, dancing through mycelium and root hairs. Here, CRISPR might one day fine-tune resilience, improve nutrient uptake, or mitigate the spread of pathogens, but only if guided by the awareness of these countless connections. Each change has the potential to ripple across unseen landscapes, touching lives you may never meet. In this quiet reflection, you sense the continuity between plants, animals, humans, and microbes. You feel your breath synchronize with the rustling leaves, your heartbeat echoing the movement of rivers, your thoughts drifting with the flocks of birds crossing the horizon. Knowledge becomes empathy, observation becomes care, and every action gains a gentle weight of consequence. You notice the interplay between intervention and observation, between thought and action, and how every small gesture can amplify across systems as vast as oceans, forests, and plains. Your gaze extends beyond immediate perception, envisioning the evolutionary timeline stretching backward and forward. You feel the ancient roots of cooperation, the quiet emergence of traits honed by millennia. You imagine how a single genetic nudge today might echo centuries into the future, shaping the dance of life in ways you cannot fully predict. Yet you also see the resilience inherent in ecosystems, the ability of life to absorb, adapt, and transform. Each ripple from your interventions may alter some paths, but the broader network carries on, offering hope, surprise, and renewal. Finally, you pause to feel the meditative rhythm of this understanding. Life is not a collection of isolated organisms, but a web of influence, dialogue, and adaptation. CRISPR is a tool in this web, a whisper rather than a shout, capable of gentle guidance if wielded with mindfulness. You envision your consciousness extending outward, touching forests, rivers, oceans, and skies, and returning with the quiet satisfaction of understanding. The threads of life shimmer faintly, as if acknowledging your presence and attention. You allow yourself to rest in this awareness, sensing that every choice, every intervention, every thoughtful pause resonates far beyond the human horizon. The world is alive in ways both visible and hidden, and your care matters, echoing softly through the concentric circles of life. As you drift in this realization, you sense the subtle guidance of wisdom waiting for the next step, a quiet invitation to explore not only life's networks but the nature of knowledge itself, and how curiosity can be both gentle and powerful. And as your mind hovers over this vast, interconnected tapestry, you begin to wonder, what is the shape of intelligence in the world beyond yourself, and how might your understanding of one small thread deepen your awareness of the whole? You move gently into the world of unseen challenges, where the familiar calm of knowledge gives way to subtle intricacies that are not immediately apparent. You notice that beneath each surface observation, hidden currents ripple quietly, carrying both promise and caution. Biology, in its nuanced complexity, does not announce its surprises loudly. It whispers, and the attentive listener is rewarded. You allow yourself to drift along these quiet waves, feeling the balance between exploration and care. Each step in this delicate landscape requires mindfulness. You watch as molecules interact with precision, yet recognize that even the most deliberate interventions can produce off-target effects, tiny shifts that may seem inconsequential at first. These hidden consequences are not failures. They are subtle reminders of the living system's depth, of the layers of complexity that make it both resilient and fragile. You learn to move slowly, observing without urgency, knowing that understanding grows in patient moments. Off-target effects appear like shadows beneath clear water, slight disturbances in patterns that otherwise seem orderly. You imagine a stone dropped in a pond. The ripples spread in ways you cannot predict fully, yet each wave follows the physics of connection and cause. You breathe with the rhythm of these ripples, feeling the gentle logic behind the unpredictability. Each unintended consequence becomes a quiet teacher, asking you to pause, to reflect, to notice what might have been overlooked. You consider the intricate dance of cellular machinery, where enzymes guide reactions with grace, yet a small misalignment can cascade outward. In the same way, in your observations, tiny misjudgments in measurement or interpretation can lead to results that differ from expectation. Yet there is a quiet beauty in this variability, a proof that life resists rigid control and invites attentive presence. You float among these thoughts, feeling a soft wonder at the orchestration of complexity around you. Challenges appear in unexpected places. A chemical interaction might ripple into metabolic pathways. A subtle genetic shift may echo across generations of cells. You witness these phenomena not with worry, but with gentle curiosity. Each unseen challenge is a lesson in humility and attentiveness. You sense that to understand the full story, you must observe not only the obvious but also the hidden, the whispered threads that tie systems together in delicate balance. In this space, science becomes a meditative practice. You do not rush to predict or control. You learn to notice, to follow patterns as they emerge, and to respect the subtle guidance of outcomes that are not fully anticipated. Even apparent mistakes or deviations are not failures. They are opportunities to see connections that were previously invisible. You lean into the quiet rhythm of this learning, feeling how each observation, each mindful pause, becomes part of a larger tapestry of understanding. You imagine yourself walking through a garden of living experiments. Each organism, each cell, is a quiet participant in a delicate ecosystem. Off-target effects are like small, hidden currents in the soil or unseen gusts of wind through the leaves, imperceptible at first, yet shaping the growth and movement of everything around them. You sense the necessity of patience, of gentle observation, and of humility in the face of these unseen forces. The unpredictability of biology carries a soft kind of wisdom. You notice how small perturbations, a slight change in temperature, an unexpected signal, a minor mutation, can alter outcomes in subtle ways. You do not try to control or eliminate them. You allow your attention to rest upon them like dew on a leaf. You feel the balance of risk and care, the interplay of intention and consequence, the harmony that arises from attentive presence rather than force. Each challenge carries its own lesson, quietly whispered to those who listen. You reflect on the concept of resilience, how systems recover, adjust, and sometimes flourish in response to subtle disruptions. You imagine this resilience as a soft, flexible lattice, absorbing pressure without breaking, bending with unseen currents while maintaining integrity. This metaphor fills you with calm, a recognition that complexity and unpredictability are not threats but companions in understanding. As you move deeper, you encounter the beauty of interconnectedness. One change, however small, can echo through networks of biological processes. You think of neurons firing in a quiet symphony, of metabolic pathways winding through cells, of ecosystems adjusting to the subtlest shifts. Each unseen challenge is a note in this symphony, a gentle nudge that reminds you of the careful attention required to move through life with understanding and care. Your mind drifts to the delicate balance of intervention. You imagine guiding a process like steering a small boat through quiet, shifting waters. Too forceful a move might upset the natural current. Too little attention, and opportunities are missed. You sense the need for gentle guidance, for observation that respects both potential and limitation. This awareness becomes a quiet meditation, each thought a wave in a continuous, soft rhythm of learning. You reflect on the quiet courage of observing without immediate answers. Science asks you to sit with uncertainty, to honor the lessons hidden in subtle deviations and gentle ripples. You feel the patience of time stretching around you, the gentle unfolding of understanding as it grows from careful observation. Each unseen challenge is a reminder to move slowly, to notice what is easily missed, and to trust in the gradual accumulation of insight. The chapter carries you further into awareness of interdependence. Systems do not exist in isolation. Even the smallest cell affects others, even the most precise experiment resonates outward. You feel a deep, gentle appreciation for this interconnectedness, for the invisible threads that bind processes together. Unseen challenges are not obstacles but invitations to see beyond the obvious, to notice the tender, subtle fabric of life. In quiet moments, you see the elegance in unpredictability. The patterns are there, though not immediately clear. The logic exists, though not rigid. You learn that each challenge is an integral part of a larger, soft unfolding. You breathe with the rhythm of these patterns, allowing the calm to settle over you while your mind remains attentive to the quiet complexity beneath the surface. And as the chapter nears its close, you sense a gentle transition forming. Each unseen challenge, each whispered lesson, is part of a continuum, a subtle guide leading you toward the next horizon of understanding. You feel the quiet pull of curiosity, the soft invitation to explore further, carrying the awareness of balance and patience into the next phase of discovery. You step quietly into the dimly lit laboratory, where the air hums with curiosity rather than urgency. Your footsteps make no sound, yet you feel the pulse of ideas vibrating softly through the room. There is a sense of waiting here, a quiet patience that surrounds every pipette, every notebook, every glowing screen. You watch his hands, steady and deliberate, move with care, preparing to interact with a world invisible to most eyes. This is the realm where Crispr was born, and you are here to witness its story. In the early days, there were fragments of curiosity. Microbiologists peering into petri dishes, studying the tiny, unassuming bacteria that carried secrets of their own survival. You can almost hear the gentle whispers of their thoughts. Why do these sequences repeat? What are they doing? Questions, soft but insistent, echo across benches and microscopes. There is no rush, only the quiet insistence of inquiry. Each day is a patient conversation with nature, a dialogue between the human mind and the elegant simplicity of bacterial defense. One of these scientists, perhaps someone like Francisco Mojica, sits hunched over a microscope. His eyes reflect not just the specimen but a growing wonder, a sense that something is there, waiting to be understood. You can feel the moment when a subtle pattern clicks. Repeated sequences, palindromes, mysterious segments flanking them, a code within a code. The realization is not thunderous. It is a soft spark that lights a path forward. You sense the excitement in the room, gentle and restrained, like ripples spreading across a still pond. Elsewhere, another scientist sketches on paper, lines of thought becoming lines on a page. Theories unfold quietly, as if each pen stroke awakens a hidden possibility. You watch as collaboration bridges continents, connecting minds across years of research. Conversations drift in the air, some tentative, some confident, all attentive to the smallest details. You imagine yourself leaning in, listening to the rhythm of discovery. Perhaps this sequence guides the scissors, or perhaps it protects the genome in ways we haven't yet imagined. The words float like feathers, light and delicate, but weighted with meaning. In one small laboratory, the first experiment begins. A cell, alive and unknowing, is chosen as the subject. You can see the careful hands introducing the Crispr system, guiding it gently like a teacher showing a student a new technique. There is no fanfare, only the soft rustle of gloves and the occasional hum of equipment. Time stretches in this space, each moment carrying anticipation and patience. You notice how the scientists watch, not with anxiety, but with curiosity, as if the cells themselves are partners in a quiet conversation. When the first edits succeed, the room does not erupt. Instead, there is a shared, almost imperceptible sigh, a quiet acknowledgement that something remarkable has been glimpsed. You sense the emotion behind the eyes, a mixture of relief, wonder, and a humble awareness that nature's patterns are more intricate than any one mind could capture. This is the human side of discovery, the patient observation, the careful thought, the subtle celebration of knowledge rather than noise. You follow another thread of the story, perhaps Jennifer Doudna and Emmanuel Charpentier at a bench somewhere, molecules and enzymes arranged like dancers ready to move. Their experiments unfold slowly, deliberately, each step a dialogue between hypothesis and reality. You can almost feel their thoughtfulness, the mental map of possibilities branching like delicate threads of light. With each test, the path becomes clearer, yet the wonder remains. What else could this tool do? How might it illuminate life in ways never imagined? In these moments, discovery feels like breathing. You are present in the quiet rhythm of curiosity, noticing the gentle cadence of thought and experiment. Failures arrive softly, teaching without judgment, guiding hands back to careful observation. Successes arrive almost imperceptibly at first, and only in hindsight do they shine like constellations in a night sky. You feel the scientist's patience mirrored in your own awareness, as if their persistence has created a space for reflection and contemplation within you. There is also joy here, subtle but undeniable. A shared glance across a lab bench, a note passed quietly, the recognition that a moment of understanding has been achieved. You sense the reverence for knowledge, the respect for each discovery, and the humility that comes from realizing how much remains unknown. Crispr is not a single flash of genius but a series of human experiences. Small, attentive, reverent moments strung together like pearls on a delicate thread. You imagine the conversations outside the lab, where ideas travel faster than light across emails and conferences, but always returning to the same core: curiosity and care. Debates unfold softly, the kind that sharpen understanding without diminishing wonder. Questions continue to ripple. Could this change medicine? Could it heal genetic diseases? Could it reveal deeper truths about life itself? The answers are not urgent. They are suggested, explored, and nurtured in a gentle dance of intellect and imagination. There are moments when the work seems invisible, yet you notice that even the quietest lab holds vibrations of history being made. You sense the echo of every prior experiment, every careful observation that made the breakthrough possible. In the rhythm of pipettes, the hum of incubators, and the slow unfolding of data, you see the story of human curiosity writ large. Crispr emerges not from a single eureka moment but from countless hours of thought, attention, and persistence. And you begin to understand the soft beauty of science in motion. It is patient, it is humble, it is alive in the everyday gestures of observation and reflection. You feel the thrill of understanding gently, a warm glow rather than a flash of lightning. It is a discovery that invites contemplation rather than applause, one that asks you to linger in wonder rather than rush to conclusion. You breathe alongside the scientists, sharing their subtle excitement, feeling the quiet pulse of revelation that spreads through their work. You see the continuum of curiosity, how small observations in a petri dish lead to profound possibilities, how experiments open doors to questions yet unasked. You sense the rhythm of inquiry and imagination moving together, intertwined, like a slow river carving its path through the landscape of knowledge. Every step of the way, human patience and intellect create a space where understanding can take root and grow. In the stories of these discoveries, you notice something gentle yet profound. The thrill of insight need not be loud. The most extraordinary moments often arrive quietly, in attentive observation, careful thought, and collaboration. You carry this awareness with you, as if the very act of witnessing these tales softens your mind and expands your sense of wonder. You recognize that the essence of science is not just in results, but in the patient, reflective journey that leads there. And as you prepare to leave the lab, you feel the story lingering, like a whisper along the corridors of your mind. The hands that guided the experiments, the minds that imagined the possibilities, the moments of revelation, all of it leaves a gentle imprint. You carry the understanding that discovery is not only about knowledge but about the quiet, human persistence that makes insight possible. You step out slowly, leaving the soft glow of curiosity behind, yet feeling its warmth within you, a bridge to the next story waiting to unfold. You sit quietly at the edge of thought, as if your mind is a page waiting for the next line. Each breath moves slowly, tracing invisible curves through your chest, expanding and contracting like a soft tide. In this quiet, you imagine your life as a manuscript, each day a sentence, each decision a word shaped with care. You notice the spaces between events, the pauses, the silences that carry as much meaning as the moments themselves. In these gaps, the future is gently written, not with urgency, but with a careful, attentive hand. You think about the rhythm of choices, how small, deliberate acts ripple outward, like a quill moving across parchment, Each decision presses lightly into the surface of what is to come. Some strokes are bold and deliberate, others feather-light, almost imperceptible, yet every mark contributes to the unfolding pattern. You sense that time is patient, allowing for revisions, for thoughtful edits, for moments of reflection before the ink dries completely. Even the unexpected, the abrupt, the unplanned, becomes part of the story, a necessary turn in the narrative that shapes the lines yet to come. As you breathe, you imagine the science of beginnings and endings. In biology, cells constantly renew, replace, and repair, orchestrating life in an unceasing, precise rhythm. Every organism is a living manuscript, written in molecules and signals, edited in real time. Your own body mirrors this pattern, repairing, adjusting, evolving in quiet harmony. Just as you revise words on a page, your cells adjust, your mind adapts, your spirit learns. The future is not a sudden chapter, but a continuous flow, like the slow unfolding of a story that you participate in with conscious intention. You allow yourself to consider the weight of curiosity and compassion in this unfolding. Curiosity is your lantern, soft and steady, illuminating corners of understanding and imagination. Compassion is the ink that traces every line with kindness, shaping interactions with care and deliberation. Together, they create sentences that feel meaningful and gentle, paragraphs that unfold without harshness, chapters that invite reflection instead of anxiety. In your mind, you see your interactions and decisions as letters in a letter to the universe, written with thoughtfulness, care, and quiet reverence for life itself. The notion of responsibility drifts into your thoughts like a feather falling slowly through still air. Responsibility is not a burden, you realize, but a form of mindfulness, a recognition that every choice affects the larger narrative. Just as a writer considers the reader, you consider the consequences of actions, the echoes of thought and gesture that extend beyond the present moment. You feel a soft weight in your chest, not heavy, but grounding, as if the knowledge that your life is part of a larger story offers gentle reassurance. Your mind begins to wander across timelines not yet written. You see possibilities stretching outward, delicate threads weaving through unseen dimensions. Some paths are illuminated clearly, others fade softly into shadow. You understand that the future is not fixed. It is a canvas for mindful shaping. You imagine yourself stepping lightly from moment to moment, tracing one line after another, aware of each impression yet free to explore new directions. Like an artist sketching shapes on a page, you move with intention, curiosity, and a quiet reverence for what is yet to unfold. In this imagined future, you notice patterns in human endeavor. Science itself is a manuscript. Experiments, hypotheses, observations, and conclusions slowly refining understanding. Knowledge accumulates, carefully edited by countless minds over generations. Each discovery is a sentence added to a vast, ongoing story, connecting past, present, and future. And just as you read and reflect, the collective human manuscript grows richer, offering guidance and inspiration to those who continue to write. You sense that you are part of this greater story, a quiet participant in a narrative that extends far beyond yourself, yet intimately connected to your own life's page. As you drift deeper into this reflection, you picture moments of gentle growth. Imagine a tree slowly expanding, each branch reaching outward, each leaf unfolding in quiet progression. Its rings record the passing years, subtle and precise, like footnotes in the larger text of existence. You feel your own life mirroring this slow, patient growth, each experience a ring, each insight a leaf of understanding. Even challenges are part of this pattern, adding depth and texture, contributing to a fuller, more nuanced story. You realize that the gentle unfolding of life requires patience, and that each day's choices are small, thoughtful strokes contributing to a masterpiece that only time can reveal. The concept of revision enters your mind as a comforting rhythm. No page is permanent, no choice irreversible. Life allows for reconsideration, for edits made with intention and awareness. Mistakes are not failures, but suggestions for refinement, gentle invitations to explore new phrasing or new understanding. You sense the soft power of humility in this process, a reminder that uncertainty is not a threat, but a space for creative, mindful engagement. You let yourself rest in the idea that even as the future is written, it remains open, receptive to thoughtfulness, curiosity, and compassion. You notice that the mind relaxes when it contemplates this form of authorship. There is no rush, no need to finish the chapter all at once. You breathe in and out, imagining words forming slowly, sentences shaping deliberately. Life is a manuscript being revised with care, each choice weighted with wonder and responsibility. You feel a quiet comfort in knowing that the unfolding story is not measured solely by speed or achievement, but by the attentiveness, care, and intention you bring to each page. Finally, your thoughts turn toward the gentle interplay between imagination and reality. The future is written not only in action, but in thought, intention, and vision. Every quiet reflection, every moment of curiosity, every act of kindness contributes to the story's trajectory. You imagine these threads intertwining, connecting the smallest gestures with the largest possibilities, forming a tapestry that is intricate, resilient, and alive. Your mind eases, embracing the soft, deliberate rhythm of participation in this unfolding story. And as you allow yourself to rest, drifting into the quiet spaces between thought and awareness, you feel the gentle certainty that the manuscript of life continues beyond the present, carrying with it the quiet echo of curiosity, compassion, and attentive care. The next chapter waits to be written, soft and unhurried, inviting you to move forward gently as the story flows onward.